it.
Okay.
Okay, you've got it.
Okay.
Perfect.
Okay, sorry about that. Alia is greatly, she appreciates the wonderful opportunities of exploring the world of research and ecology in Davis and the surrounding area. Along with her studies, Alia is also an artist, musician, an aspiring educator, and overall nature lover. Ian is a second-year PhD student in the UC Davis Animal Behavior Graduate Group. He is interested in the role of behavior in wild birds' responses to challenges caused by produced by humans, as well as ways the behavior research can inform bird conservation, especially within urban environments. Its focus is on the challenges of chemical pollution, and it's exploring ways to bridge the fields of ectotoxicology and behavioral ecology in his research. Working with the dapper Black Phoebe and its nestlings, Ian is planning to pick apart relationships between an individual's environment, behavior, reproductive success, and risk of exposure to chemical pollutants. Ian is thrilled to be doing work here where he gets to think about and interact with the local birds he loves every day, and is infinitely grateful that he is no longer spending so much time in windowless laboratories like when he was an undergrad studying neuroscience at UCLA. Outside of his graduate student life, Ian is an illustrator, a climber, and I assume a proud bird dad to two adorable budgies. So, excuse me. Ian will begin tonight's talk. He will introduce his graduate research on the exposure of Black Phoebes to heavy pollutants. Alia will share results from her undergraduate research project on risk perception and reproductive success in Black Phoebes. So with that, I turn it over to you, Ian.
Thank you so much, Ken. Sorry for, I guess, hamming it up with our introductions. But as you'll see over the course of our next hour together, Alia and I are very fun people. We're huge nerds for birds and for Black Phoebes especially. And so we really hope that you enjoy our talk. All right. All right, Ken, how does this look?
Looks great. I can see the starting slide.
Okay, wonderful. All right, so getting started. Hi, everyone. As Ken mentioned, Alia and I are here to represent Project Phoebe, which is a just wonderful program that was started by myself and other graduate students that I'll introduce you to in a second here at UC Davis. And our focus is on understanding how wild birds respond to novel and rapidly changing environmental conditions. Largely, we focus on urbanization as a major force of environmental change, but we think about other factors of change as well, and we work both in urban and non-urban environments, as we'll talk about. And then something to orient you to here on this first title slide is the bird featured in that little illustration there. That's the Black Phoebe. That'll be the star of our show. And so if you learn nothing else during this hour together, you're going to learn how wonderful these birds are. And there he is in the bottom left. All right. So once again, we are just part of the broader Project Phoebe team, and we're here representing the team as the very grateful recipients of support from the YOLO Audubon Society. And so again, my name's Ian, and I'm a second-year animal behavior PhD student, and then later you'll be hearing from Alia, who is one of the members of our first team of undergraduate researchers that we were able to hire this past year. This is our full team. So we accomplished so much, as you'll see, during our first field season, and it wouldn't have been possible without each and every single one of these people. Having, honestly, like a whole family put together to answer the questions that we were all interested in was such a great experience. and it was what enabled us to accomplish so much in our few months of field research. In particular, on this slide of our team members, I want to highlight Sage Madden, who is second from the left in this photo. Sage is a third-year ecology PhD student, and it was Sage's idea to study Black Phoebes. And then very graciously, she opened up her research world to more and more of us as we also fell in love with these birds and with the questions that they can help us answer. So to give you a sense of our roadmap for this evening, we'll be talking about first Project Phoebe as a whole. So even though we are just two members, we are very excited to tell you about what Project Phoebe is, the unifying themes that it seeks to explore, what we accomplished in general during our first field season, which recently concluded, and then in turn, we'll be talking about our sub-projects, the types of questions and answers that are closest to our hearts. So at that point, Alia will be switching off with me to first talk about her research project that she conducted. And so you'll hear about whether flighty birds are less successful in cities. And then I'll come back in to tell you about my research with chemical pollution and Black Phoebes, which, just as a silly little title, I've called Bugs for Lunch with a Side of Pollution. If you're anything like me, that doesn't sound like a very appetizing lunch, and we'll learn more about that at that point. And then we'll hopefully save some time for questions. That being said, if you have any questions while we're talking, and you're not sure if you'll be able to stick around until the end, please feel free to write things in the chat. And then when I switch off with Alia, I'll be looking through and seeing what kinds of things people are curious about, and I'd be happy to answer your questions in the chat. All right. So to start off with Project Phoebe as a whole, the objectives that unify our work are generally that we seek to understand what features of cities and human-altered habitats challenge birds or help them thrive. And we do this using the Black Phoebe as our star, as our focal bird that we research. And the Black Phoebe is a small insect-eating songbird. Many of you are probably familiar with the Black Phoebe. That's been one of the most wonderful parts of our work, is that even when we think that people won't have any idea what we're talking about, birders and non-birders alike are often familiar with this bird. I think that's because they're our neighbors, for sure. They share our urban and suburban spaces, and also because they're so charismatic and so noticeable. So I talked about how we're interested in these cities and human-altered landscapes, and these landscapes can be challenging places to live for birds especially, or for birds certainly, as well as for other wildlife. And some of the things that can make these places challenging are higher temperatures, different predator communities. For example, many of you are aware of the impacts of outdoor cats on wild bird communities, as well as higher or different concentrations of chemical pollutants. So all of these things that come along with us and with our activities as humans. That being said, some species, but not all species, persist and even thrive in environments that have been altered. And that really makes us and many other biologists interested in the question of how. How are they doing well, despite all the challenges? So just some examples. We're all familiar with some very robust and successful urban birds like the house finch, these birds that use our resources, that share space with us in cities and don't seem to be going anywhere anytime soon, despite the challenges, as well as non-avian urban animals that have stabilized in cities, might even be spreading like fox squirrels. So this leads us to some questions, again, focusing on the Black Phoebe specifically. We were curious about whether Black Phoebes are thriving in cities. Are they one of these successful urban animals? And also, knowing that Black Phoebes still live in quote unquote natural habitat and not just in cities, we wondered, is nearby natural habitat better for them than cities, despite being human altered as well, despite having its own collection of impacts? And then from that, what are the specific habitat features that promote or prevent Phoebe success in these different types of environments? And lastly, um, many of us who work on this project are really interested in animal behavior. For some of us, it's like our bread and butter. And so we also wonder, do behavioral changes allow Black Phoebes to cope with these human challenges? So is that part of their toolkit for responding to change and to challenges? So getting into what our project really looks like I mentioned, the star is the Black Phoebe, and to give you a little bit more natural history about this species, um, starting really basic, the Black Phoebe is a small black and white flycatcher, and historically it was found near rivers, streams, but increasingly we find them in human-dominated habitats, uh, like cities and suburbs. And the two things that I want everyone to know about Black Phoebe ecology are that in terms of their foraging, these birds are aerial insectivores. So they forage in a way that's called sallying, where they'll fly from perch to perch and snatch pretty large insects right out of the air. So different foraging style from other insectivores like warblers, which might pick insects off of leaves and twigs and stuff like that. They're like aerial hunters in a way. And then also these birds build mud cup nests, so that distinguishes them from a lot of other birds whose nests we might be more familiar with. You can see an example of a Black Phoebe nest on the bottom left there. So on the inside, it looks like a nest you might be familiar with. They line it with plant materials and then drop their eggs inside. But that lining is being held by a very meticulously made cup of mud and straw. And this enables them to put their nests on things like vertical walls, and then historically on things like tree cavities, indentations in riverbanks, and sometimes even rock faces. Now, we mostly find these birds nesting on vertical surfaces on human-made structures like our houses, barns, bridges, etc. All right, and so, sorry, the setting for our work is what we call the urban gradient. So as I mentioned, we're interested in the range of experiences that this species has to cope with across the different environments that it can be found in our area. And so doing urban ecology for us means studying urban environments, natural environments, and everything in between. We really want to capture this full spectrum of environmental contexts. So here on the bottom left, you can see that you might find us doing our work on a busy intersection, like here at the end of B Street and Russell in Davis. And there's me with a Phoebe in hand. That Phoebe tried to nest right there uh, and would regularly fly across that busy intersection. But then we also study Phoebes in maybe they're more like ancestral type habitat, like here on the right in riparian areas and wetlands. And so there's me checking a Phoebe nest that was constructed at Cache Creek Nature Preserve out by Woodland. And then what our activities look like as we go about our field season come from the world of avian ecology. So we use some fundamental techniques like mist netting there on the left, where we use nets to very gently and respectfully be able to have these birds in hand. And then once we have these birds in hand, that enables us to do things like band them. So there's Alia in the center, banding a nestling, Black Phoebe. We band both adults and nestlings. And that enables us to, of course, keep track of who's who thereafter, even just through binoculars. And then lastly, our project, all of our projects that we perform as part of the overall Project Phoebe rely heavily on nest monitoring to give us a sense of how Black Phoebes are actually doing. Are their nests successful? How are their little babies doing? So we use a variety of techniques like these telescoping mirrors or just getting up there on ladders to check on the contents of all of the nests that we monitor. And once they reach a nestling stage, we'll regularly give those little nestlings, like you see there in my hand, a checkup. Another very important part of what we do is thinking about the context in which these nests are found. And so this entails a lot of measuring of the environment. So we'll take direct habitat measures. This entails things like thinking about how much of the area immediately around the nest is made up of water, is made up of native plants, is made up of invasive plants, is made up of asphalt or pavement. What is the average height of the tree canopy in the area? These kinds of variables give us a sense of uh, the broader environmental conditions there. And then we narrow in on some specific environmental conditions using technology like in the center pane, we have a thermometer placed near a nest. So across all of our nests, we place thermometers, and this helps us understand what kinds of thermal environments are being experienced at different nests. The data coming out from this, as Sage could tell you with the analyses that she's been running, are pretty shocking. And even just while we were on the ground, we had temperatures experienced at some Phoebe nests that were as high as like 125 degrees Fahrenheit. It's not easy being a bird in the Central Valley in the summer. And then the kinds of environments that Black Phoebes nest in, especially in urban environments, can get extra hot. And then lastly, a way that we measure aspects of the environment is through measuring chemical pollution. And this is a bit interesting. I'll talk more about this later in my talk, but as you can see there, I'm collecting feathers from the Phoebe in my hand. So the way that we measure this aspect of the environment is indirectly through biological samples from the birds themselves. And I'll talk about how that's possible at the end of the talk. Lastly, I mentioned that our bread and butter as a team is the study of behavior. So some of the ways that we get at the behavior of these really awesome birds are conducting foraging follows. So there on the left, you see a rough estimate of the foraging range of a young fledgling. There's a not very good picture of that fledgling. That's an inset on the bottom left. If you look closely, you can see that he's pretty cute. But we follow these birds and make sure that we're just focusing on the same bird for the period of time. We measure what kinds of things they're foraging on. Are they foraging on the ground? Are they foraging in the air, as we would expect? And what is their foraging range? Additionally, we had the amazing ability to place GoPros at pretty much all of our nests this past season. And that enables us to get pretty good resolution video. A screenshot of such video is shown here in the center, where we can see a playback of the interactions between adult Black Phoebes and their nestlings. So again, this is a screenshot from a video like that, and you see here one of our banded females visiting her nest. And if you look closely, you can see that one of her nestlings is actively begging, hoping that he or she will be the one chosen to get that food delivery. And this lets us get an understanding in detail of, again, these interactions between adults and nestlings and characterize the quality of parental care that nestlings are receiving. Lastly, Alia will be talking about a cool behavioral technique where we measure the flight initiation distance of birds. And I won't get into that because Alia is our specialist when it comes to this kind of behavioral work. So now I would love to share with you all some results from our 2023 field season. As I mentioned, we were interested in the most basic sense in like, how are these birds doing? Are they doing better in cities? Are they doing better in riparian areas? And what kinds of factors in each environment might be causing them to do well or to do poorly? So here are some results. During our 2023 field season, we were able to monitor 49 nesting attempts, and 47 of these were right here in Yolo County. So our work is very local. We're very much enmeshed in this community, and we care a lot about the birds immediately around us. If you're wondering where the other two were, they were in Sacramento County. We are hoping to do some more work this coming field season in the Sacramento area. And this is a map of all of the nests that we monitored. So each of these little red blips was a nest that was active. So that means that birds were at least trying to nest in it. An active nest can have eggs, it can have nestlings, it can have successful fledglings, depending on what stages it makes. So we worked at Cache Creek. If you follow the stars, you'll see the areas that I'm talking about. We worked at Cache Creek as one of our natural areas. We worked in the city of Woodland as one of our urban areas. We worked in Davis, where you can see we had a lot of success finding nests. We worked along Puda Creek at various access points, again, providing us with another example of a natural area. We worked at the Yolo Bypass, providing us an interesting example of a natural area. We worked in the city of West Sacramento. And then lastly, we worked along the American River Parkway. And I personally was so interested in the American River Parkway as a study site because it is a natural area. For those of you who have biked or walked along the lower American River, you know how much of like a greenway it is, how much of a healthy riparian corridor it is in some areas especially. But it's also right there in the middle of an urban, you know, an urban landscape. So I'm so interested in doing more work there. We're excited to have some partnerships with the Sacramento County Regional Parks Department to think more about the experiences of birds specifically along this like simultaneously urban and natural area. So as I said, we monitored 49 nesting attempts across those locations that I mentioned, and 28 of these attempts successfully fledged adorable chicks like the one on the right there. Yippee! Of course, that means that 21 of the nesting attempts failed completely at some stage. So failing completely means that they did not fledge a single chick. Whereas the 28 others did fledge at least one chick. Often, if they fledged any chicks, they were fledging around 3 or 4. And this works out to a 42% failure rate for the nests monitored in our first season. And when we break this down and we look at categories of nests at a basic level, we find that nests that were built in highly urban areas were the most likely to fail. So looking at the map on the right, I've color-coded nests that failed, that did not fledge any chicks, as black, and I've color-coded nests that were successful as red blips. So the nests that were in highly urban areas are places here, like in Central Davis, near Central Park. And the leading causes of these failures were things like lethal temperatures. I mentioned how hot some of our nests got into the 120, 130 degree range, as well as scrub jay predation specifically. And this was really interesting to us that it seems the biggest predator impact to nestling Black Phoebes is scrub jays. We were thinking maybe something like Cooper's hawks. We were thinking a lot about house cats and stuff like that. In contrast, when we take the opposite end of the spectrum and we think about highly non-urban areas, so this is like no buildings around, maybe you have a creek nearby, these nests were the least likely to fail, and only 25% of nests in this type of environment failed. So as an example, you can think of somewhere here, we were monitoring a handful of nests along Puda Creek near Old Davis Road that were successful. I want to emphasize though, first of all, this is preliminary and it's not statistical, but it is motivating us to look a lot more into the challenges faced in urban environments specifically. But we still recognize that nests in non-urban areas, even these nests along Puda Creek, are still impacted by human challenges. So I mentioned that at the very beginning. Human alteration of the environment comes in many shapes and sizes, and one of those is just urbanization. Usually urban environments come with many other types of effects of humans, but we can find those impacts in quote unquote natural areas as well. So some of the challenges that birds might face, that our Black Phoebes might face, even in these highly non-urban areas, are things like limited nest sites and chemical pollution. So if any of you are aware of the geography of Superfund sites here in Davis, you might know that there's actually a Superfund site, a designated EPA Superfund site right here off of Old Davis Road where those successful nests were. So we have reason to believe that that's a highly polluted area, and it could be impacting the birds in ways beyond just whether they successfully fledged. And that's one of the things that I'll talk about later. But yeah, I really want to emphasize here that whether we're talking about urban environments or non-urban environments, it's important to think about the challenges that are uniquely presented in each of those and ways that we can mitigate them. Yes, and so we have more nuanced outcome analyses to work on. We're looking at things as well as nestling growth rates and impacts to nestlings in the egg stage specifically, behavioral impacts, and other kinds of sublethal effects. All right, so beyond how the nests did, I wanted to share some of the other dimensions of our first field season. And one of the things that was really rewarding for each of us involved in Project Phoebe this past year was community engagement. It was immensely enjoyable for me to be doing an urban ecology project, and especially one that was so local, because of the opportunities it provided to interact with just people in Davis, people in Woodland, and then communicate with them about how wonderful our urban birds are. So we worked with homeowners this past field season to monitor nests that we wouldn't otherwise be able to. And homeowners found their participation to be a rewarding experience. And of course, this was wonderful for us because we found that the majority of urban phoebes seem to nest on homes, actually. So this gave us another outlet to find more phoebes and study more phoebes in the areas where we worked beyond just the public parks and the handful of structures that are present in public parks. And I want to take a moment here to point out that we are looking to recruit even more homeowners to join the cause of studying and supporting the Black Phoebe this coming spring. Like I mentioned, this was such a critical part of the flow and the outcomes of our study. And we had really great success in Davis. So for any listeners, especially who are coming from Woodland or other areas of Yolo County, please do keep us in mind if you see a Black Phoebe hanging out around your house, even this time of year. And then please do contact us. I'll share contact information at the end if that bird ends up building a nest and you would feel comfortable with us coming by to study the bird. Oftentimes, this comes with opportunities to, for example, hold nestlings and to really get a strong sense of how birds are doing in your backyard, which I think is really gratifying. Additionally, we had a really great time developing flyers, brochures, and even a website to reach homeowners, as well as just to teach the broader public about our feathered neighbors. And we were even featured in a few local newspapers, which was awesome. So word really got out about what we were doing, and it made us so happy that that was the case. And yeah, community engagement is so important to me, to Sage as well. Our interactions with folks here in Davis and beyond mean the world to us. So if you have any more ideas about how we can engage with more community members, especially in places like Woodland and West Sac, where we weren't able to reach quite as many people as in Davis, please let us know. And you can find our information as well as just in general, learn more about what we're doing and get updates at our website, projectphoebe.com. One last mention of the first Project Phoebe field season that was wonderful was mentorship. So we mentored 5 undergraduate students and we provided them with a local and accessible field research experience. And coming from a non-field biology undergraduate experience, I just thought that this was the most amazing thing to be able to, still take your classes and not have your schedule totally disrupted, not have to like go backpacking out in like Alaska or something in order to have a field experience, but just to like get picked up by you know, a grad student and go get these experiences. And so our undergraduate researchers develop skills like wildlife handling, banding, and collection of biological samples, including some of them even became pretty proficient at safely bleeding wild birds to collect samples for DNA analysis and other analyses. And then also each of our 5 students designed and completed an independent project. So you see an example here of one of our students, Farah. So all of our students gave a presentation to our group based on something that they conceived using data that they took initiative to collect. We helped them analyze the data in R and help them put together presentations so that they could have the full scientific experience and not just be, you know, hands helping us do the work. And with that, oh, so here are all of our students. We had great talks about the impacts of noise pollution on our Phoebes, the impacts of chemical pollution in general on our Phoebes. Again, more noise pollution, the effects of urbanization on the shape of Phoebe bills, which was super interesting. And then lastly, we had Alia, who put together a really wonderful presentation. So I will now turn things over to Alia to present her research.
Thank you, Ian. If you give me a few minutes here, Alia, excuse me. I failed to restart your bio when I started the recording, so I would like to get that on the recording. And so if you give me a few seconds, I will do that. And those who heard the bio at the beginning, please bear with me. And this is for the recording and those who came in later. Okay. Alia is a third-year grad student, undergrad student at the University of California, Davis, majoring in mathematics and the minor in wildlife and fisheries biology. She's excited about working in the field, observing bird behavior and learning more about the scientific process and how birds and humans can coexist in an increasingly urbanized world. Alia greatly appreciates the many wonderful opportunities for exploring the world of research and ecology in Davis and the surrounding area. Along with her studies, Alia is also an artist, a musician, an inspiring educator, and an overall nature lover. So please welcome Alia to our program tonight. Thank you.
Awesome. Thank you so much, Ken. And as you just heard, I was super excited to be a part of this. I just came across Sage giving a presentation during like a bird watching club event, and I'm like, oh my goodness, I want to be involved. This sounds like so much fun and so much learning, and that's exactly what it was. And so my sub-project I'm calling, or the title is, Are Flighty Birds Less Successful in Cities? And so in particular, I was interested in bird behavior, as Ian was talking about before, and how humans and wildlife can coexist in many of the same spaces. So as we talked about before, but just to recap, rapid urbanization is a major threat to avian biodiversity. In cities, there's often a lot less kinds of birds, different species of birds, and altered resource availability and higher levels of human activity can pose big challenges for birds in urban environments and reduce species diversity. However, some birds, like these pigeons here, doing really well and even like benefiting from easy access to food that people drop and stuff like that. And so there's those pigeons as an example, but there are also other birds that seem to be populating human or like urban areas more, or so it seems. but then we still have a very limited understanding of the mechanisms that enable some species to survive and live in changing human-altered environments. So my project aims to gain understanding of these ways that birds respond, and one of the ways is through behavior, and behavior is perhaps one of the more immediate and more flexible ways that birds respond in changing environments. So one of the ways that birds respond is that has been studied in urban in accordance to, or like in relation to urbanization, is flight initiation distance, which is the distance at which a bird flushes when approached by a possible threat such as a human. And most studies have found that birds in less urban areas have higher flight initiation distance than birds in urban areas. And that could be because birds have developed increased human tolerance to humans, and because it is also a energetically costly escape is also an energetically costly. Birds that are around humans more may have developed increased tolerance and maybe don't even view humans as predators in urban areas. Another behavior that I was really interested in when thinking about Black Phoebes in particular was their sort of telltale tail flicking behavior, that I'm sure if you've noticed Phoebes, it's almost just a part of like, oh yes, that's a Phoebe, is this bobbing of their tail that they do quite often, like almost all the time. And so I was looking into it, and I found not very much information on why they do this, but what I did find was that tail flicking behavior in Black Phoebes is thought to signal awareness to potential predators. So like if a hungry Cooper's hawk, for example, was looking for a next meal, maybe they would choose a like less active target than a like very vigilant looking, quick and agile Black Phoebe that was like bouncing its tail a lot. So in this way, tail flicking behavior might be a predictor of risk perception and energy allocation, overall like fitness of the bird. However, studies do suggest lower foraging success as well with increased tail flicking. So it does have its costs. Another interesting behavior that I will look into more. And so just to come back to a little bit of why the Black Phoebe is especially interesting to study these escape and like risk perception behaviors, is because they are seemingly abundant in urban areas. Like I'm sure almost everyone has seen a Black Phoebe like today, or heard them. They are very well known by birders and non-birders, as Ian sort of said earlier, and they often also return to the same spots for foraging, for singing, and also for nesting. So they are very familiar to many people. But how much do Black Phoebes actually benefit from living in urban areas? And I will try to gain some insight into that through these next things. So my main research questions were, number one, does flight initiation distance, or FID, and tail flicking rate in Black Phoebes differ across an urban gradient? And consistent with previous studies, I expected less urban birds to have longer FIDs and higher rates of tail flicking than more urban birds. And then my second question has to do with how, like the consequences of having a different FID and tail flicking rate in different environments. So does the relationship between FID and tail flicking rate and reproductive success differ in birds across an urban gradient? I predicted that, oh, too fast. That in urban birds, higher FID and tail flick rate would be associated with lower reproductive success, since they would be spending more time like always getting scared away, and less time, leaving less time for them to forage and care for their babies. While in rural and like with less urban birds, having a higher FID and tail flick rate would be associated with higher reproductive success or just less bad consequences for reproductive success than in urban birds, because they would be appropriately keeping themselves safe in order to take care of their babies and themselves. And then my last question that I focus my project on is, what is the relationship between FID and tail flicking in Black Phoebes? Sort of like, do they get at the same effects or meaning for the Black Phoebes and risk perception, or what is going on between them? So my predictions are that increased FID would be correlated with increased tail flicking rate and vice versa. So how I answered these questions, along with the fieldwork and like all the huge amounts of different kinds of data we, as a whole team, whole project collected, these ones are what I focused on specifically. I used a semi-automated program to create an urbanization index. So I used this program and trained it to recognize different surfaces, I guess, on a Google Maps image, and then turn that into a number. And that's how we got the range of urbanization that our sites were in. So stuff like paved surfaces, trees, houses, other vegetation, it took into account. And then for FID, measured using a rangefinder by sort of just walking straight towards the bird and then measuring, stopping and measuring where we were in relation to it when it flew away. And then counted tail flicks by just watching the bird, and tallying the amount of tail movements it made, while also timing it with a stopwatch. So what do we find? The first question, does FID differ across an urban gradient? Just to orient everyone to this graph, we've got the urbanization index along the x-axis, with the higher number representing more urban, and then flight initiation distance along the y-axis. And what I found was higher urbanization index is associated with shorter FID across all breeding stages. So that aligns with previous results on urbanization and flight initiation distance in birds. And we can see that here with, it looks like birds with eggs were the least, took the longest to flush when we approached them, and then those with nestlings were especially flighty. So I think that's very interesting. And also, even after taking into account urbanization, when we measured individual birds multiple times for FID, there was no consistency. So you can, like, a certain bird didn't have like its own specific FID, which is also very interesting. It seemed very flexible and depended on just the urbanization. And so given this, this trend, this relationship, is there, like, what is the benefit to being flighty, more flighty in urban environments? And doing the same thing with tail flicking rate. So now we still have urbanization on the x-axis, and now we're looking at the tail flicking rate. There was not a variation across the urbanization gradient, but there was a significant effect of whether the bird was banded or not on how much it flicked its tail. So we're not exactly sure why. It could be because there could be an effect of banding on tail flicking rate, like birds that were not banded had a higher tail flicking rate than birds that were banded. Or maybe birds that are less vigilant were more likely to get caught by us than banded. I thought that was just a very interesting result that came up. And so now on to the relationship between FID and reproductive success. So we know that FID changes along the urban gradient, but does it affect reproductive success? What I found is that FID is not associated with nestling mass or nestling survival, which are two ways we are measuring reproductive success. And we can see that with this graph. So on the x-axis, now we have FID, because we're looking at that in relation to nestling mass on the y-axis. And each of these colored lines are different urbanization index levels. And the shading shows the uncertainty. So there's a bunch of overlap, as we can see, like the greenish shading goes almost all the way up and down towards the right side. So there isn't enough strong evidence to show a relationship here, but there may be other effects of FID variance across an urban gradient, perhaps such as adult fat scores that maybe we will look into later. So that was interesting. And now with tail flicking rate, however, there does seem to be a trend of decreased nestling mass as tail flick rate increases. So here we have a similar graph just with tail flick rate on the x-axis, and there is some overlap here in the shading, but much less than in the flight initiation distance one just before. And the slight relationship seems to exist only with nestling mass and not nestling survival. So that's what's pictured here with the effect on nestling mass, which that is also interesting. And their tail flick rate is, or the relationships between tail flick and nestling mass or survival are not statistically different across urbanization indices. So although it doesn't change tail flicking behavior, it doesn't seem to change based on urbanization. It does suggest an effect on nestling mass. Also, I want to mention that low sample size may explain a lack of effect. So hoping to get more data as the project does more seasons. And then my last question was, what is the relationship between FID and tail flicking in Black Phoebes? And the data show that there's a weak positive correlation between FID and tail flicking rate. You can see here we got the average FID and then the average tail flicks, and it's not super strong, but there may be something going on there that show that both behaviors could be some indicator of risk level and perception by these wonderful birds. So are flighty birds less successful in cities? We do know that birds in cities are less flighty. So you can keep in mind, like, if you're at Puda Creek and there's a Phoebe that you think is nesting, like, they'll likely fly away or like flush at a farther distance than maybe the one that's on your neighbor's house or your house. And so what we found so far is that birds in less urban areas will flush around, like, sort of greater than 15 meters away. And urban birds are more in the like 10 to 12, 10 meter range. And then we do need more data to make robust conclusions about some trends, especially with reproductive success, and sort of what's going on with that. And if we're not seeing reproductive success consequences as a result of flight initiation, like being flighty or less flighty in different environments, like what else is going on? Maybe there are other costs. So stay tuned, and I will now hand it back to Ian to talk about his part of the project.
All right. Hi again, everyone. Thanks so much for your attention as we walk you through these different parts of the world of Black Phoebes. And as one final destination in our talk, I'm going to be telling you about the experience of these birds with chemical pollution in our region. And we'll be talking about how they eat bugs for lunch, but often that comes with a side of pollution. And you can imagine that is not a great diet. So before I get into that, I'd like to talk about how chemical pollution in general is a complex crisis that affects all of us. And to illustrate that, I put together this little piece here, which shows, across different environments that we might find ourselves, whether we're out on Puda Creek, whether we're walking around town, or whether, we're thinking about agricultural contexts. Both humans and wildlife are being exposed constantly in this day and age to a variety of chemical pollutants. Right, so all of the environments that we occupy, we share those with wildlife. And so we share the consequences of this pollution. However, historically, there's been a lot more attention paid to the effects of chemical pollution on, the human population. So the public health aspect of the chemical pollution crisis, and that's incredibly important, but we also need to bridge what's a bit of a species gap and think more about how these compounds are affecting all kinds of animals, not just human animals. And so that's where this kind of work comes in. Sorry, Ken mentioned at the beginning that I consider myself an ecotoxicologist, and that means applying the world of toxicology, the study of, environmental toxicants, to their effects on the whole ecosystem and to wildlife. So when we think about birds and when we think about Black Phoebes specifically, I can share with you that we found that chemical pollution does affect the outcomes of nesting Black Phoebes. Um, this analysis was performed by one of our wonderful undergraduate researchers, Joanna Rose. And just to orient you first to the relationship that we're thinking about, if you take a look at the graph on the right, on the x-axis, we have a measure of like pollution in general in the environment. So this is like a pollution percentile. It's like if somebody gave that place on Earth, that nesting environment, a score or a grade for how badly polluted it is, 100% being really, really polluted, 0% being perfectly pristine. And then on the y-axis, we have the probability that birds have a successful fledging experience. So this means that at least getting one of their little nestlings safely out of the nest and onto the next stage of their life. So up at the top there, one would be, yep, absolutely, you will at least fledge one nestling from your nest. 0 being like, there's no chance at all, your nest is certainly going to fail. And we see here that as we progress from these really pristine environments to more and more polluted environments, the probability of, being a successful parent, having successful fledging decreases, and it drops off pretty fast as we get to these like extraordinarily polluted environments. So to summarize, exposure to polluted environments overall is negative for our Black Phoebe's nest outcomes. And now I want to tell you about where this pollution percentile, this like pollution grade, this score comes from. It comes from a wonderful publicly available data set called Cal EnviroScreen, which is an effort of the state of California to assess how polluted different regions of the state are down to like a very fine geographic resolution. So this is a database that the public can explore. You can open it up and you can see a view of how polluted your neighborhood in Davis is compared to other neighborhoods, compared to Woodland, compared to Bakersfield. They have really good data for the whole state. But the numbers that come out of this take into account all kinds of different types of pollution, like air pollution, heavy metal pollution, neonicotinoid pesticide pollution, and so on. So this really is just a complex mix of chemicals, and therefore also a complex mix of the effects of those chemicals that we're seeing when we think about this relationship between pollution and Black Phoebe fledging success. This is where my research comes in. And so what I am seeking to do as a graduate student here, beyond just like the great, wonderful world of Black Phoebes in general, is to isolate specific chemicals that might be affecting the Black Phoebe and understand those specific chemicals' effects. So that instead of working with a complex mix of, things going on, we can really start to understand like what might be driving this relationship between polluted environments and decreased nesting success. And so following my understanding of Black Phoebe natural history and also the history of pollution in our county and in the Sacramento region in general, I honed in on a compound called methyl mercury, which I'll be abbreviating as MEHG. It is a heavy metal pollutant that's found in our region. So people often also discuss this as mercury pollution. You might have been exposed, no pun intended, to some information about mercury pollution in our area. But it's more accurate to say that you get exposed to methyl mercury than to mercury per se. So that's why I have this distinction here. Methyl mercury is one of the World Health Organization's top 10 pollutants of concern from a public health perspective. It is very detrimental for human beings to be exposed to methylmercury. So one of the places you might have come across information about methylmercury is if you are a fisherman or a fisher person, you might see signage about what is safe to eat and how much, right? And the goal of this is to reduce your exposure to methylmercury. Additionally, methylmercury is known to affect wildlife. Historically, the focus on research understanding methylmercury levels in wildlife has been with fish. Going back to part of the reason that I just mentioned, we want to know how contaminated fish are in case people are eating them, and also in water birds like waterfowl, especially also penguins, which is super interesting because these birds consume a lot of fish, way more fish than probably any of us do, and so you can imagine they're most obviously at risk to be affected by methylmercury. And one thing that I would like everyone to know about methylmercury is that it, what we call bioaccumulates and also biomagnifies through the diet of animals. So a basic kind of cascade of methylmercury is that the compound starts out in water and then it gets taken up by algae. The algae are technically what make it methylmercury and not just mercury. And once it's in that state in the algae, then it can be transferred to other organisms and start affecting them. So you might have tiny zooplankton eat the algae. When they do, they build up all that mercury inside of them. A small fish might eat those zooplankton, building up a lot of mercury inside them as they age and as they continue to eat and eat. And then larger fish will eat those smaller fish and have just eventually overwhelming levels of mercury. And this is why you're discouraged from eating, these large fish that are higher up in the food chain. So the point here is that as an animal continues to live and as it continues to eat, if its environment is contaminated with methylmercury, the impacts can compound. They can get worse and worse over time. Now, when we think about our region, this was so fascinating for me to learn about when I was starting to do this work. I had no idea about these kinds of histories in the Sacramento area, but historically, mercury mines were run in the Coast Range. So if you think out along the Blue Ridge, the Copiah Valley, those mountain ranges, and that has led to the leaching of mercury into watersheds there and into waterways. And then additionally, when we think about the Sierra Nevada, we think about a very long and important history of gold mining, very famous history of gold mining. Gold mines required the use of mercury as a kind of factor in the refinement of gold. So mercury would be mined in the Coast Ranges, polluting the environment there, and then it would be transported to the Sierra Nevada to assist with the gold mining and refinement, where it would get into the soil there as well and leach into the water. So what this leaves us with in the present day is that anytime it rains, essentially, some amount of mercury from both directions, both to the west of us and to the east of us here in the Central Valley, enters the watershed and then is transported to areas downstream, so to our region, and it ends up in the lower parts of our creeks like Puda Creek and Cache Creek, the Lower American River, as well as in our very precious and expansive wetlands here in the Central Valley. So this is some data from a YOLO bypass symposium that was presented in 2015. So these numbers are a little bit out of date, but this group, McCord Environmental, was presenting on the levels of methylmercury in our area. And what I want you to see is that this arrow, the green arrow pointing to 0.06, those are kind of like the levels we want. Those are levels that are pretty safe for humans and wildlife alike. But where we are is up by that red area. So looking up at that red area, that's getting towards Yolo County, getting towards the Yolo bypass. And at this time, the levels were many times over what we want. So we're in the red zone. And when I started learning about this, I was like, huh, phoebes generally maintain a close association with aquatic environments, and it seems like our aquatic environments are polluted with methyl mercury. So I wonder if this is one of the most impactful chemicals that they are exposed to. So my first question in conducting this research has been, what affects the exposure of black phoebes? As I mentioned, exposure to this heavy metal happens through dietary intake. And so if you think about a black phoebe's place in the food chain, um, you might imagine that it all starts out again with little zooplankton. Those are the little blips on the far left. Those might get eaten by a small insect, something like a caddisfly or whatever in an aquatic environment, which is in turn eaten by a predatory insect like a dragonfly or by a small minnow. And then those are the things that we find or expect to be consumed by black phoebes in their traditional aquatic, their creek and river environment. And so they are multiple steps up the food chain, and that puts them at risk of being detrimentally exposed to methyl mercury if that methyl mercury is present in the water. However, I became really interested in the possibility that not all phoebes are equally likely to be exposed to methyl mercury. When we think about the behavior of individual birds, differences between birds and the way that they forage, will lead them to eat different things. And they also have different levels of interaction with the urban environment that would altogether shape their risk of exposure to methyl mercury. So for example, one thing that we started noticing very early on in our fieldwork was that black phoebes don't always forage in the way that you read about in a field guide. So they're not always sallying. They're not always catching these larger insects or skimming things off of the surface of the water. Sometimes they are foraging right off the ground in more upland areas, even when there's a creek nearby. And so in such a context, for a phoebe that has a different foraging style and therefore a different diet, you might have a food web like this, where there's just like a direct link between a phoebe and something like an earthworm. This is something that we have observed them consuming. And that removes the phoebe pretty much entirely from the aquatic food web, and therefore from potentially the risk of exposure to methyl mercury, which is cycling within that aquatic system. And then once I answer this question of which phoebes are exposed and what affects how, exposed to this problematic compound they are, I also, of course, want to answer the question, what's the harm? So if a bird is exposed at a certain level, how is that affecting them? And approaching that in a way that's like meaningful to their ecology. So one thing that we know from public health studies and from studies with captive non-human animals is that methyl mercury is a neurotoxic compound, even at low levels of exposure. So if you are exposed to extraordinarily high levels of methyl mercury, it can start causing really serious problems, potentially like even organ failure. But even at low levels, it's been found that it can affect your brain cells. It has these neurotoxic properties where it can affect the way that brain cells communicate. It can cause the death of certain brain tissues, neural tissues. And in humans, this leads to potential cognitive and behavioral effects. And coming from a neuroscience background, I was always interested in what about the cognitive and behavioral experiences of other animals, not just humans. So if black phoebes are exposed to methyl mercury, even at low levels, well, they have brains too, of course, just like we do. Um, and they might be experiencing these neurotoxic consequences of exposure. So I'm wondering whether exposure to methyl mercury impairs the cognition of black phoebes and decreases their performance for important behaviors like parental care, which could be something that leads to reproductive success. You would imagine if you don't have the mental, cognitive resources to coordinate with your partner and provide really high quality parental care day after day after day while you're raising nestlings, that could lead to reduced food delivery, reduced quality of food, which would impact the experience of your nestlings. So this project is in progress. I guess that's the way that graduate student work goes. It's complicated and multidisciplinary, but I'm happy to share that part of the data collection for this effort to answer those two questions is complete. So first, we have started conducting and have conducted many surveys of foraging behavior in our birds. As I mentioned, we were able to follow individual birds around in the field and observe them carefully, capturing their foraging style and what kind of substrates they were using. So we score every bird on every single foraging attempt that it makes. How high off the ground was it foraging? Was it foraging over water, over grass, over asphalt, etc.? Did it land in order to forage? Did it sally? Those kinds of things. And in so doing, we've seen some really weird foraging behaviors, like Black Phoebes who like to forage on asphalt in more urban areas, and will hop around looking kind of like robins or thrushes, finding worms in little dirt piles in parking lots. So their diet is a lot more complicated than we might have expected before doing this research, just from reading a field guide. And I'm super interested in how that affects their experience of the world and of pollution. Additionally, we completed a lot of recordings of parental care. So if you think back to the very beginning of our presentation, I talked to you about how we put up GoPro cameras at our nests, and this enabled us to record multiple hours of footage for every nest of the interactions between parents and their nestlings, as well as interactions between parents and other parents. So some of the behaviors that we can observe through this footage include handoffs from one parent to another of food, as well as the alternation of parents performing food delivery. These kinds of behaviors, I believe, indicate some level of complex coordination between adults in order to, reach that optimal delivery of parental care. So I'm really interested in quantifying those and characterizing those types of behaviors to give each nest like a score of how coordinated its parents are. I believe that if you have, some kind of neurotoxic effect that you're experiencing, it would be harder to perform these more coordinated efforts with your partner. All right, and so the reason that I can't present to you the end-all be-all results at this time is because there are two aspects of data collection that are still in progress. So these are very exciting to me. The first is measuring the diet of Black Phoebes. We have completed a process of collecting hundreds, and I mean hundreds of poop samples from nestlings. We have so many vials full of these weird little fecal sacs. I never realized how strange the poops of insectivorous birds might be. But if you have like a parrot, or if you have seen bird droppings out by your feeder, they look nothing like that. They're very squishy and weird. So we have all those samples in hand, and we're planning to perform what's called stable isotope analysis on these, which will help us understand how much of the material is made up of aquatic prey versus terrestrial prey. And so that'll help me understand like where an individual family of Phoebes is sitting in that set of food webs. Are they sitting more on the aquatic interaction side, or are they maybe the type that's like foraging on the ground, eating lots of earthworms and stuff? And we can pair this with the foraging behavior that we've measured for the parents of these nestlings. And then the most important type of data collection, which is starting in 2 weeks, is quantifying the exposure of all of our Phoebes, both adults and nestlings, to methylmercury. So the way that I am going to do this is using their feathers. We've collected, again, hundreds of feathers from both adults and nestlings, and these are sitting in a dark and not wet and sterile environment awaiting mercury quantification, which the results are going to be cool. The process is not super, you know, cool in any way. It's going to be me returning to my life of being in a laboratory and just running samples. But I am so excited to see the numbers come out of that. It'll be using a machine that looks a bit like the one shown in that picture, where I fill a little glass boat with chopped up Phoebe feathers. And then the machine incinerates those. And in the process, it can look at how much of um, the feather that it had just incinerated was methyl mercury. All right. And so why do I think that this type of research matters? And why do I think that it'll be really exciting to get those methyl mercury results and start looking at the exposure of these birds to the compound? At a very broad level, I want my work to direct attention to non-lethal effects of pollution on wildlife. As I mentioned, in general, there's a species gap between understanding the effects of pollution on humans and on wildlife. And a big part of that species gap is that we haven't gone very far beyond understanding the effects of pollution, beyond just like saying, this is killing wild animals. You know, this is a lethal dose, or this is something that's enough to make the eggs fail, or what have you. But there are a lot of things, a lot of detrimental effects that can happen before you ever get to that lethal dose. Like I mentioned, there can even be behavioral effects. Additionally, I hope that my work helps to determine if exposure to pollution in wildlife can be predicted by an individual animal's behavior. So I hope that this can be done, because one of the challenges in studying the effects of pollution on wildlife is there are so many different species, there are so many different chemicals to think about, um, you know, and there's so many different areas on our planet now that are polluted with various chemicals that it's hard to make priorities and hard to allocate the time to monitoring whether certain animals are exposed and monitoring the consequences of that. But if we can more simply monitor an animal's behavior and then determine, okay, these animals are more likely to be exposed to this compound because they have this type of foraging behavior or because they have this type of nesting behavior, then we can, you know, have some shortcuts for really studying the groups that need our attention most. Yeah, so again, this gives us new opportunities for risk monitoring, which is one of the most important aspects of wildlife toxicology. Additionally, I want to demonstrate through this work that our region's remaining natural areas are just as impacted as they are important. So in no way can I claim that Fuda Creek and Cache Creek and the Lower American River and these different type of sites, the Yolo bypass, are like not helpful for birds, not important for birds. They are so important, and it is for that reason that I want to identify some of the ways that they are still impacted and promote mitigation of these effects so that these highly important habitats can be more completely beneficial to the wildlife, to the birds that utilize them and depend on them. And lastly, I think this work is really cool because it highlights the ingenuity of birds that are coping with environmental change. This is kind of like a recurring theme across our research, but in this case, it's been so gratifying to observe what are basically like novel behaviors in the Black Phoebe as it's striving to use different environments, right? So I mentioned like foraging in parking lots and changing the types of insects that they consume, the types of invertebrates that they consume. I don't have the video, but we even had someone send us a video of a Black Phoebe who forages on cat food, like dry cat food that's left out on their patio. So these birds are incredibly clever and they're incredibly resourceful in the ways that they cope with environmental change. Novel behaviors, that's what it's all about. All right, and so with that conclusion, I want to thank everyone here. I want to thank Alia as well for helping me give this presentation and share about Black Phoebes. If you're wondering what this image is, it is an image of a banded adult who is being weighed. But we always get such a kick out of this part of the data collection process because we use, and trust me, it is a very standard thing to use. We use a little cut of PVC pipe to help keep the birds stabilized and calm while we're weighing them. But it means that we get an up close view of little Phoebe butts whenever we're taking their measurements. All right, so just as a last note, once again, I encourage you to visit our website. We do our best to post regular updates there, and we also provide some general information about the world of urban ecology, about urban birds and urban wildlife there. So that's www.projectphoebe.com. And then please do keep in touch if you have any ideas about where to find more Phoebe nests, what kinds of things we might be missing in our studies of Black Phoebes, or if you want to share experiences that you've had with urban birds, you can reach me, at ighaliburton@ucdavis.edu. And now, for the folks who are able to stick around, we're happy to take questions either through the chat or I'm not sure if you're able to speak up and ask.
Excuse me. You know, we had some questions in the chat earlier, and I noticed that we do have some folks who are on a audio connection only, so they don't have access to reading questions in the chat. You think we have time? I think we might have some time to read some of those questions, but there are two new questions at the bottom, I think. No, those are thank yous. Let's see. Oh boy. So, Ian, you had answered a question from Kelly Davidson in the chat. Do you mind going through that for folks who don't have access to the chat?
Yeah, definitely. Kelly asked us about what Phoebes might do in the event that a nest fails. So I mentioned that many nests unfortunately did fail. And she was wondering, will Black Phoebes lay a second clutch in the same nest? And are they ever able to double clutch after successfully fledging a first clutch? So this entails successfully raising, a group of like 3 to 4 babies and then doing the whole thing again. And the answer is that yes, Phoebes will often try to keep using the same mud cup, even if they've failed using it. This is something that kind of distressed us. For example, we had one bird who kept trying to nest on the underside of a bridge, and it was building its nest on a piece of styrofoam, which was so unstable, and the nest would fall, and the bird would insist on rebuilding the nest in exactly the same place. Additionally, we had cases where a nest might have been really hot and a bird could have potentially found like a better shaded area or whatever, but then they continued to use that nest even if it didn't seem like a good idea to do so. But then at the same time, we had some birds who were perfectly successful the first time using a nest, but then all of a sudden decided that they didn't like it anymore, I guess, and would build one sometimes even just like a foot away or like on the opposite wall of the house or something like that. So, it's something that we would constantly talk about in our team. Like, what is it that leads them to make these decisions? How do they decide where to build their nest? How do they decide whether to reuse a nest or, put in all this work to build a new one from scratch? And then additionally, regarding the question of whether they can fledge multiple broods within a single season, we definitely did see that. For our successful birds, they tended to have upwards of like 10 nestlings within a season because they would have multiple successful broods.
Okay. Sage had responded to Kelly on another question about Kelly had asked how could she encourage Phoebes on her property. And Sage, are you still on?
I can also answer
Okay.
that.
Yeah, I
Okay.
can just it over to Ian. I just wanted to answer in case folks are
Okay,
popping off,
okay.
but I'll let Ian take.
Yeah,
Right.
so that's a really good question. For anyone who's still on and listening, if you care about these birds like we do, if you think that they're really cool and really cute like we do, you might be wondering, how do I encourage them to come around my property? We're still, as Sage mentioned in the chat, thinking about, or not thinking about, but like actually measuring all of the different things that might be important to them and important to their success. But before we get into like the nitty gritty of those kinds of analyses, I would just say like think about their natural history. What they really depend on is bugs and mud, if that makes sense. So if your property has features that promote insect, like, healthy insect abundance and diversity, obviously we're not talking about like mosquitoes. I'm not encouraging anyone to create mosquito habitat, but if you have like a variety of native plants, potentially if you have some kind of like water feature, like I've seen a lot of front yards in Davis that have little like kind of sloughs or little indentations in the yard to collect rainwater. Those kinds of things will all promote insect abundance and diversity, which provides easier foraging for Black Phoebes. And then if you have some source of mud nearby, then they'll really benefit from that. A last note about that, something that I didn't share today because it's going to be one of our new initiatives in the second field season that's coming up, is we're looking at ways that you can like directly provide a nesting site for Black Phoebes. This isn't really an issue in urban environments because pretty much any house, any building, they can find somewhere to nest on that works for them. But in theory, if you maybe live out on a bigger property, like some kind of ranch, and you have a lot of open space, if you want to provide a nesting site for Phoebes, there's something similar to a nest box called a nest shelf that you can build and put out for them. And so we're going to be piloting that to see if Black Phoebes will use those in this coming field season.
Okay. Thanks. Erin wants to know, she wants to know if drought affects foraging behavior through learning from parents.
Let me think about that. So if drought affects foraging behavior through learning from parents, I definitely, just to start here, we definitely get the sense that like how wet a year is has major impacts on like the experience of this species, no doubt on many other bird species as well. But for example, our first field season here was conducted in like what I've heard was one of the wettest years that Davis has experienced in a while. And I would imagine that that was largely beneficial for foraging. It had some of its pros and cons in other ways. So it seemed like Phoebes had to wait longer in order to start nesting because of all of the intense weather. But once the weather cleared up, it seems like they were buffered from some of the heat early on, although it got really hot later. And it did seem like there was a good diversity of insects. So yeah, that's kind of like a wishy-washy way to answer, but I do believe that how wet a year is has big effects on the availability of insects to forage on. And how that affects the nestlings, we definitely observed that fledglings stay close to their parents for many days after leaving the nest until eventually the parents will start yelling at them and kind of like ask them to leave. So there's a long period of time where they could be learning from the parents while they're out of the nest, maybe even while they're in the nest. So I could see all of those things being connected for sure.
Okay. Excuse me. Lynette is curious about the nest failure by high temperatures. What does it look like? Was it that the chicks literally die of heat, or how did you tell?
Yeah, I am glad you asked. It's really hard for me to like think about this. It has affected me to see this because depending on what stage the nest is, it takes different effects. So we had instances where a nest that was still on eggs, the eggs just never hatched. And when we held them up, to a light, they looked kind of weird. They seemed more opaque than they should have been. And so we got the sense that they might have, like, to some extent actually cooked, like in the sense that the proteins might have denatured in the heat, and then the eggs had to be abandoned. It's even sadder once a nest reaches nestlings. If a nest gets too hot, then the babies have no control over their thermal environment. Obviously, they're in the nest. The only recourse that they have is to leave the nest to try to get to a cooler place, but they are in no position to leave the nest. And so it could cause like forced fledging, or I think more accurately, it seems like when it gets really hot, the individuals in the brood will try to get farther away from each other to reduce like that transfer of heat, from like their collective body heat. And there's only so far away that they can get from each other in that small nest cup. So they end up falling out of the nest and injuring themselves.
Okay. Wow. All right. Looks like the last question. Melinda wants to know if did you mention how far the phoebes will forage from their nest? What's their range?
Yeah. So the range of a phoebe, like the general territory of a phoebe in the literature, was estimated to be around like max 2 football fields.
Wow.
Yeah, which is pretty big, but like not super big if you think about it. And generally phoebes are thought to like stay close to the ground at all times. They're non-migratory. Um, they usually are described to be foraging like 6 to 10 feet off the ground, and they perform short flights generally. So all those things help make sense of why they wouldn't have a super huge range. But we found that really depended on the individual and might also have something to do with urbanization and other kind of like habitat features. So, for example, I mentioned I was super interested in the Black Phoebes along the American River specifically, because these are birds that are living in an urban environment but still have access to a waterway, to a more natural area. And so for some of those birds, we were finding nests built in neighborhoods that like abutted the American River Parkway. So the nest would be on a building in a neighborhood, but then I would watch adult birds fly upwards of like half a mile at a time in order
Wow.
to travel from that house, from that neighborhood, to the bank of the American River, because they still relied on that river or preferred to go to that river because it provided, I believe, higher quality foraging and an abundance of, mud and other nesting materials. So I really do think that it depends on what they have access to. And that was one of my motivations for, thinking about that birds who have immediate access and immediate proximity to a waterway, to a creek or a river, probably have a much easier time of like raising young than other birds.
Okay, great. Thank you. Okay, so thank you very much for this talk tonight. I found it very interesting because I'm kind of partial to neighborhood Black Phoebe, so I'm glad that we had you guys as speakers. Yeah. Anne, do you have any parting words for tonight? Closing words?
Coming. Oops.
Caught you by surprise, sorry.
Oh, whoops, gotta close my door. Well, I would just like to thank both our speakers and say I'm really excited to follow your research as you get farther along, and I hope you will come back and speak to us again, and also consider applying for more funding from our small grants program if it seems appropriate and you have a need to see. It's just so much fun to see ideas germinate. you're still in the early days, and it's going to be wonderful to see what happens. So thank you.
Okay.
And
Yes,
I just
thank you.
also want to, in fact, Ken should be saying this. Ken, I just wanted to mention the next meeting and getting
Oh,
ready.
go, please.
Yeah.
Oh, I'm doing it?
Should I?
Okay,
Our next meeting that Ken's in charge of is December 6th, and it'll be our annual Christmas bird count identification meeting and organizational meeting for picking out where you, the members, want to, and friends want to, how you want to participate in the bird count and what areas. We're divided into 10 different areas around Puda Creek, and with a radius around that, and you'll be learning about each of those areas. You'll also be learning a little bit more about eBird and how to use it, because that's how we prefer to collect our data now. And we'll be having some fun ID questions on different species that may be tricky IDs in the area. So please come December 6th, and it will also be, Ken, am I right, our first in-person meeting?
Correct. At the Davis Senior Center, 646 8th Street. The address and everything will be in the publicity for the event.
Yep. Great. All right.
Okay. All right. Hey, thank you everyone and Ian and Alia, thank you so much for tonight. I really appreciate it. I thank everyone for joining us tonight and those hanging in there to the end, because there were some great questions and answers. So everyone, I hope you have a good rest of your evening, and we'll see you next time around. Thank you so much.
Thanks, Ken.
All right. Good night, folks.